Gyrolab immunoassay solutions have many applications, including testing for SARS-CoV-2 antibody and for pharmacokinetic analysis of monoclonal antibody therapies. There's a need for reliable serology immunoassays detecting anti-COVID-19 antibodies in human serum. Sensitive antibody detection is needed for a wide range of serum antibody levels in COVID-19 patients. High throughput capabilities are also needed for projects screening large numbers of samples. Advantages such as low reagent volumes minimize project costs. Rapid assay times provide essential results-driven decisions, and robust and reproducible results are required for immunoassays in regulatory environments. Gyrolab immunoassays are available for qualitative analysis of antibodies against COVID-19 in human serum and for pharmacokinetic analysis of therapeutic antibody development. Gyrolab miniaturized microfluidic CD-based immunoassays dramatically reduce the sample and reagent volumes with a rapid time to data of about 1 hour. The immunoassays are fully automated with minimal hands-on time and are designed for 21 CFR Part 11 compliance in regulated environments. The Gyrolab COVID-19 serology immunoassay is available for the qualitative detection of total antibodies generated against the spike protein receptor binding domain, RBD, of SARS-CoV-2 in human serum samples. The bridging immunoassay format consists of a biotinylated RBD His as capture reagent and Alexa 647 labeled RBD FC detection reagent. Reagents and samples are placed into wells of a microplate, loaded onto the Gyrolab platform. Then the immunoassay is completed automatically with data available for 112 data points in about an hour. The specificity of the immunoassay to antibodies against SARS-CoV-2 was evaluated by analyzing serum samples from 50 individuals collected pre-pandemic in comparison to negative and positive controls. The pre-pandemic serum samples all tested within the same range as negative controls, with an assay response for serum samples spiked with positive controls also in the expected ranges. The Gyrolab assay produced 112 data points in an hour. Specificity was also shown by inhibition of the assay response, where various amounts of unlabeled RBD protein was added to 1 microgram per ml of a positive control. The assay samples were in neat serum. The response of the positive control was effectively inhibited by the unlabeled RBD protein, confirming assay specificity. The SARS-CoV-2 antibody assay showed a broad dynamic range from 40 nanograms per ml to 200 micrograms per ml. This bar graph shows assay responses for a negative control and positive control samples of increasing concentrations of rabbit polyclonal anti-SARS-CoV-2 spike RBD antibody in human serum, diluted 1:4 in Rexup-H buffer. The assay was used to analyze 42 samples that had been confirmed positive for IgG or IgM from a lateral flow immunoassay, with 41 of these samples appearing positive using the Gyrolab antibody assay. Gyrolab SARS-CoV-2 antibody immunoassay saves samples, saves reagent volumes, saves time, and provides a broad assay dynamic range. Only 4 microliters of sample is required per data point compared to 100 microliters for ELISA analysis. 100 micrograms of capture reagent will produce about 112,000 data points, and 100 micrograms of detection reagent about 134,000 data points, stretching reagents to produce more data points per dollar spent. 112 data points are produced in about an hour, and the broad assay range of 40 nanograms per ml to 200 micrograms per ml means less sample dilutions are required for samples with high antibody levels. This type of Gyrolab SARS-CoV-2 serology test can be used in many applications in ongoing public health efforts, including inclusion or exclusion criteria for clinical studies, seroprevalence studies of COVID-19 immune responses, and healthcare worker screening. The anti-SARS-CoV-2 antibody test can also be used in the development of effective vaccines and CLIA lab tests. The ready-to-use and robust Gyrolab SARS-CoV-2 immunoassay protocol specifies the assay design, CD type, antibody capture and detection reagents, Rexit buffer, dilutions, and needle wash buffers. The protocol available on the Gyros Protein Technologies website saves development time and reduces reagent use. Gyrolab is an open system and can be used for other SARS-CoV-2 antibody immunoassay formats. Antibody therapeutics development for COVID-19 includes interest in repurposing approved cytokine inhibitors to prevent or dampen the hyperinflammation seen in patients infected with COVID-19. Among the FDA-approved interleukin inhibitors being investigated for repurposing, Actemra is currently in phase 3 clinical trials for the prevention of acute inflammatory response. The Actemra Gyrolab PK assay protocol can accelerate bioanalysis in IL-6 inhibitor clinical development. Ask us about Gyrolab PK assays for your antibody therapeutic development project to save time and sample.